All right, I think we'll get started. First of all, I'd like to welcome everyone to the C-DEBI Network Speaker Series. Today, we have the pleasure of having Dr. Janine Ashe give us a talk today. Janine earned her PhD in geochemistry from UCLA in 2017. While she was at UCLA, she worked on some samples from IODP, in particular IODP Expedition 347, and that was the Baltic Sea paleoenvironment leg. While she was there, she worked with clumped isotopes. That's isotopes of carbon and oxygen in various forms, and that served as a nice foundation for her work as a postdoctoral fellow. As a postdoctoral fellow, right now she's currently at Rice University, and as a postdoctoral fellow, she submitted a proposal to C-DEBI and was funded as a postdoctoral fellow in 2017. The title of that funding is Tracing Deep Biosphere Methane Cycling with Clumped Isotopes. And today, she's going to expand upon that and other things related to clumped isotopes. So I know with the online aspect, if you have questions or comments, you could type them in as we go. She may or may not see them, but we'll definitely get to them at the end. And with that, I welcome Janine and have at it.
Okay, thank you, Jeff, for the introduction, and thank you again for the invitation to give the talk, and for those of you who are joining today in the channel. So on my first slide here, before I move away, I have this long list of collaborators at the bottom, kind of an arbitrary alphabetical order, but I do want to shout out to Stephanie Berger in particular, who basically taught me everything I know about keeping methanogens alive in bottles. And also Edward Young, who continues to allow me to measure clumped isotopes of methane on a very fancy machine I'm going to tell you about. So most seminars that I've ever been to begin with coffee and cookies. So we're going to sort of virtually pause here to get our coffee and cookies. But I also want to use this as a metaphor. So in your seminar at your department, maybe half an hour before, coffee and cookies are set out, you go, you grab a couple, you get your coffee the way you like it. It's a nice, way to be collegial in your department, catch up with people you don't see every day, for instance, ask, oh, how are the kids? How's that band you're in? And so on and so forth, right? But what if your departmental policies were different when it came to coffee and cookies? So for instance, maybe 30 minutes before the seminar, you're all going to line up outside the room, and then the doors are going to unlock at precisely 30 minutes before, and everyone's going to run in and get coffee and cookies, and you're just going to grab the things that are closest to you. So if you were first in line that day, good job you get all the Oreos, but if you're farthest from the door, you might be stuck with just the crumbled broken bits of oatmeal raisin cookie and the dregs of the decaf, right? So that's not a very fair system. So maybe your department chair would utilize something different. He or she would say, we're going to walk in the room and everyone is going to roll this pair of dice, and you're going to be randomly assigned a type of cookie and a type of coffee based on the number you roll. So in this scenario, everyone kind of has an equal chance to get what they like, but you have just as much of a chance to get something you don't like still. All right, and so finally, sort of a third scenario we could think of is a negotiation one, right? You're all going to walk into the coffee and cookies area, and before anyone takes anything, you're going to have a conversation. So, Jeff and I might both be eyeing, the pile of chocolate chip cookies, as well as coffee with cream, but maybe there's not enough to go around, so we're going to have a friendly conversation. And sort of do some trading back and forth until everyone in the room or in your department has got something they like for coffee and cookies, but maybe you weren't able to get everything you like. All right, and you can imagine, no matter which one of these policies were implemented in your department, that they would leave sort of a fingerprint, right? This kind of stuff is going to be coming up in faculty meeting, or perhaps even, in the office of your ombuds person, whether or not your coffee and cookies is a free-for-all or a negotiation or a random. And so, of course, I'm not really spending all this time talking about coffee and cookies. I think of processes as chemical reactions, which also leave fingerprints, okay? And so these metaphors that I have down here are kind of related to chemical reactions, which we tend to think of in terms of whether or not they're kinetic, so whether they're happening quickly and irreversibly, whether they're happening in equilibrium, so perhaps there's some back and forth, whether it's with a molecular species or isotopes, and then stochastic, which is not in and of itself a type of chemical reaction, but it's kind of a convenient framework to measure from. And so I use molecules with more than one heavy isotope to trace these types of reactions. And so what you're seeing here are two molecules of methane. They both have a cardinal mass of 18, and they both have two rare isotopes on them, although those isotopes are different. So we have 13CH3D and 12CH2D2. Even though they have the same cardinal mass, when we get down to the atomic level, it is a little bit different for each of them because the isotopic composition is different. And when I measure these in the lab, I measure them relative to a stochastic or a random abundance. So that's this big delta that you see here, kind of representing a departure from the stochastic, whether the composition I'm looking at has more or fewer of these rare molecules that I would expect. Okay, and so using isotopes to trace chemical reactions isn't new. We've been doing this with stable isotopes for 70 some years. And in each case, we have some substrate. It undergoes a process or a chemical reaction, and we end up with a product. And the product is usually what I, the isotope geochemist, measure. Unfortunately, with stable isotopes, really both the substrate and the process are sort of going to end up in this product. And those can be kind of difficult to pull apart. So to show you sort of how that works in action, I'm going to show you this plot here where I have sort of the bulk isotopic composition of methane both in carbon on the x-axis and in hydrogen on the y-axis. And what you can see are all these tiny little dots represent environmental samples. And so methane that's made microbially tends to utilize the light isotopes of both carbon and hydrogen. Biological reactions tend to use the lighter isotopes. They're more favored in these reactions, and they leave behind the heavy isotopes. Thermogenesis is another way we could get methane, so breaking down organic molecules through pressure and temperature. But what they're breaking down, of course, are the bodies of things like these microbes. So it's a little bit heavier because there's sort of a fractionation between these two. And then finally, we could be making methane abiotically at a range of temperatures and a range of different Earth environments. And so here the carbon isotopes are going to be the heaviest, but we have a really wide range of hydrogen isotopes. Methane often gets its hydrogen isotopes from water, and you can see there's a really wide range of water reservoirs on Earth. So you can kind of see some of the problems with using this method, because not only is the process important, but also the substrate is important. So where the starting material is coming from and what isotopic composition it has. All right, and then we could also sort of add an additional layer of complexity here, in that we could oxidize these pools of methane and change them. So here's sort of a pathway for microbial oxidation. So I could take methane that was created through this methyl atrophic pathway, oxidize it somewhat microbially, and move what remains into something that's looking like abiotic methane. So me, an isotope geochemist, comes along and I measure this methane here, and I think, well, it looks in this abiotic pool. How would I know it's undergone this process without a lot of other supporting information that I might not have? Okay, so how are clumped isotopes different, right? We still have a substrate. It's still going to undergo a process. We're going to end up with a product that I get to measure. What's different is that the important thing here is really the process and not the substrate. And that's because when we measure these molecules that have more than one rare isotope, what we're really looking at is that bonding chemistry, that process. And the substrate turns out to be a lot less important in that instance. Okay, and of course these processes that we're going to look at are chemical reactions that are happening both kinetically and in equilibrium. And then I'm going to use that stochastic framework to kind of figure out where we are relative to one another there. Okay, so a similar diagram in that I've got my two isotopologues of methane plotted here against one another, 13 CH3D on the bottom, 12 CH2D2 on the y-axis. And this black line here represents thermodynamic equilibrium. So that means methane that's made in an equilibrium reaction is going to end up on this line at the temperature that reaction occurred at. Okay? So that's going to be really straightforward. Only things that land on this line were formed strictly in equilibrium. All right, but there's all of the space outside the line, and there's a lot of ways we could get there. For instance, we could be mixing between two different pools of methane that have different isotope reservoirs associated with them. We could be diffusing. So if this was our starting gas here, the residue would be evolving in this way to the left, while the diffused gas would be evolving in the other direction in a one-to-one relationship between these two isotopologues. Or I could have a kinetic reaction, which is going to land us somewhere below the line here. And finally, I could oxidize. So this would be like the type of oxidation that's happening in our atmosphere where methane is consumed with a chlorine or a hydroxyl radical. Okay, so this gives you a little bit of idea of how a diagram like this might work in clumped isotope space. And I'm going to spend the rest of the time here talking about other processes that we can sort of assign regions to on a diagram like this. But before I move any further, I'll just give you a little bit more background. So I want to make it clear that I measure these molecules at natural abundance. So these experiments are not using spiked or labeled isotopes. These are at their natural environmental abundances. So let's talk a little bit about how rare they are. So if everyone on Earth was a methane molecule, right, there'd be about 7.5 billion of us. About 83 million of us, or equivalent to the population of Germany, would be 13CH4. 4.6 million of us, like the people who reside in the capital of Egypt, Alexandria, would be 12CH3D. Only 52,000 of us would be 13CH3D, and you would get to be my neighbor in Montrose, Texas, and all of the methane molecules here really like to eat tacos. And then finally, 12CH2D2 is the rarest molecule that we measure. There's only 1,000 methane people on Earth, equivalent to the population that overwinter in Antarctica, would be this 12CH2D2. Okay, so in order to measure things that are so rare, we need this special instrument. It's called the New Instruments Panorama. It is a high-resolution gas source isotope ratio mass spectrometer. And I love talking with microbiologists because your technology moves so quickly. Things are always changing because you have a lot of industry partners who are really interested in what you do. And so a lot of you guys, you don't even bother specializing in a particular piece of equipment. Stable isotope geochemistry is not like that at all. So this instrument is part of sort of a class of instruments that were developed in the last decade that represent a big fundamental shift in the way that we measure stable isotopes. So we really hadn't made a whole lot of progress since the 1950s in our mass spectrometers. And so this machine and others like it are able to basically do orders of magnitude better than we ever have been before. So this is why we always talk about this, and we're really excited about this method. So the way isotope ratio mass spectrometers work is you stick your methane in here. It goes through this complicated bit called the electronic sector. There's all types of plates in there that are sort of directing that stream of molecules in a vacuum. It gets to this really big magnet. It weighs about 3.5 tons. It took this thing behind it, which is an engine crane, an engine lift, to get it up here. And the magnet separates all the molecules of different mass, and they finally end up down here, either in a traditional collector for the more abundant molecules, or in this big long fancy tube, which is an ion counter. So people always like to ask me, how much methane do you need? And so these are all of the molecules that we're measuring for clumped isotopes right now, and these are the amounts in case that is of interest to anyone. Okay, so now I'm sort of going to talk about more about filling out that diagram in the different zones. So we're going to start with how are methane molecules made, and I've already let the cat out of the bag on this one. They're made abiotically, thermogenically, and microbially. So I'm going to cover those first two really briefly, and then spend the majority of the time talking about microbial methanogenesis. So here is where abiotic methane ends up on a diagram like this. Two different types of experiments. One is silane decomposition. So this is like a big molecule that you heat it up and it releases methane. And when you do this at high temperatures, like 600 degrees, it seems to be doing this in equilibrium, right? We're here on the black line. But as the temperature of these experiments gets lower and lower, we start to get further and further away from this thermodynamic equilibrium line. When the temperature gets low enough, it's hard to get this reaction to go forward at all. And so we start to change to a different type of abiotic methane formation, that which is made through the Sabatier reactions that happen at these lower temperatures. And what you can see here is sort of the overall trend is that the lower the temperature of these types of reactions is, the further out of equilibrium we are, particularly here on the y-axis. And so we hypothesized this to be due to hydrogen tunneling, and this is kind of just a fancy way of saying the lower the temperature is, the harder it is to get two deuteriums on a single molecule of methane. Is sort of the easiest way to think about that, although I'm sure if there are any physical chemists in the audience, they would have words with me. Okay, so let's move on to thermogenic methane formation. So there are actually two data points here, but they overlap, so it's kind of hard to distinguish between them. They're from the Marcellus and the Utica shales, which are some of our natural gas reservoirs here in the United States. They're basically on the thermodynamic equilibrium line at about 170 degrees, which is right in the middle of the gas window, sort of that the range of temperatures at which organic matter breaks down into methane. And that is consistent with the closure temperature of these basins. Okay, so we have that. So let's go ahead and move on to the fun stuff, the microbial methane formation. And I'm going to look at the lens of methanogenesis sort of through the lens of what this enzyme that's doing here, methyl coenzyme M reductase, or MCR. And this is the last enzyme that a molecule of methane sees before it leaves the cell. And there are several methanogenesis pathways, sort of the three classic ones that get a lot of attention: hydrogen atrophy, taking CO2 and hydrogen, making methane as a byproduct; methyl atrophy, taking a methyl group, producing hydrogen as a byproduct; and then acetoclastic, which I'm not going to talk about any further today because I don't have any data from it. But then there are also some other newer methanogenesis metabolisms that I am going to talk about some more. One is methanomycelicoccus style. It says in quotations. I don't think this has like a formal name yet, but methanomycelicoccus is from sort of the newly described 7th order of methanogens. You probably all brought methanomycelicoccus with you today. It's one of the archaea that are actually in the human gut. They're also in the guts of things like termites and other ruminants, but they've also been found in both brackish water and marine sediments. So potentially this is a metabolism that we don't really know how important it is globally. It's different from these two because it's eating a methyl group, but it has to do that in the presence of hydrogen. It can't just break down a methyl group by itself without hydrogen, and it also can't break down CO2 even in the presence of hydrogen. And then the other one I'll talk about briefly is methoxydatrophy. So these are the coal eaters. They're eating these complex molecules associated with coal deposits and producing methane as a byproduct. Okay, so as I thought about what's going to control isotope pairings or the clumped isotope composition of these different pathways that methanogens have, there were some options. So the first big one, of course, is the metabolism itself. Will the metabolic pathways be distinguishable on this diagram that I've been showing? But another option is the organism itself. So there could be vital effects, right? Will Methanosarcisa mazii always produce the same isotope pairings, even if we give it different foods, or not? And then temperature. If metabolisms are equilibrating, then I should expect to see a relationship with temperature. And then finally, the rate of reaction. So if these metabolisms range from kinetic to equilibrating, then I should expect a relationship with rate. So I will first show you all of the data we have. These are all xenic cultures of methanogens grown in lab conditions. And so here they all are. We fondly call this the microbial array. I am going to take away now those weird newer metabolisms, the methoxydatrophy and the methanomycelicoccus, and we're going to be left with these two groupings. One is hydrogenatrophy, and the other is the methyl atrophy. Okay, and so you can see here there's sort of two things that stand out to me as I look at this. One is that all of the methane produced by these exenic cultures of methanogens was below this equilibrium line. None of it is in equilibrium. It's all down here in this kinetic space. And then, of course, the second thing that you could see is that not only are these distinguishable, but there's sort of a pattern here, right? The methylatrophic ones seem to be further out of equilibrium, whereas these hydrogenatrophic methanogenesis methane seems to be a little bit closer to equilibrium. So why is it in disequilibrium, and why is methylatrophy more disequilibrated than hydrogenatrophy? And let's look at the enzymatic pathway, and I'll tell you what I'm thinking about this. Again, so here on the left is the proposed pathway for hydrogenatrophic methanogenesis. So we start down here with our CO2. Hydrogens get added at various steps in this process. We make it up here to the top. We have our enzyme MCR. A final hydrogen gets added. Methane goes on its way. So what's actually happening here? When we start adding these hydrogens on one at a time, there is some experimental evidence, a paper from Dave Valentine, that suggests some of these steps are actually partially or fully reversible. So we could put a hydrogen on, and it could exchange its isotopes with the water that's hanging around in the cell, for instance. And maybe that sort of thing is happening not just at one of these steps, but at 2 to all 3 of them. Okay, so we could be partially equilibrating a methyl molecule down here. So by the time we get up here, we have this CH3COM molecule. It sees MCR. There's some machinery that's going to add on that last hydrogen. But when we add on that last hydrogen, methane is a waste product, right? There is no benefit to keeping that methane hanging out on that enzyme and catalyzing a hydrogen isotope exchange, for instance. Like, let's get it out of the cell and on its way so that I can make my next methane. Okay, so this is sort of a potential way that hydrogenatrophy could be partially equilibrated, but when we get to that final hydrogen addition, we're just going to slap a hydrogen on, get it out of the cell as quickly as possible. All right, and so that could explain why it is still disequilibrated, but maybe closer to the equilibrium line than it otherwise would be if none of these exchanges were happening. So then let's take a look at methylatrophy. So methylatrophy is different in that it inherits sort of this preformed methyl group, right? It's just eating whatever methyl group it was that I fed it. That doesn't really have anything to do with the environment. It doesn't have the opportunity to go through any of this machinery that could sort of exchange its hydrogen isotopes with. So we're basically bringing in something that's probably already not in equilibrium with the environment. And then again, it sees this MCR, slaps a hydrogen on its waste, get it out of the cell as quickly as possible. So sort of to sum that up here, the final hydrogen addition in methanogenesis seems to be non-reversible. And so this would cause the disequilibrium in clumped isotopes that we both observe in cultures and potentially in environments. Okay, so let's think about some of the other hypotheses that I had. What if I had the same organisms, but I fed them a different food? All right, so these are 3 pairs you're seeing here. Here's the first one. This is Methanosarcina mazii, which I have given TMA, which corresponds to this point here, and MEOH, so they're about 20 per mil different and 12 CH2D2, definitely not the same clumped isotope composition. This one up here is Methanomycelicoccus luminensis, first grown on TMA plus H2, then grown on methanol from H2. These again, also very different, about 40 per mil in 12 CH2D2 and a couple per mil in 13 CH3D different. And then finally, I have M. shangliensis. This is one of the coal eaters eating one of these complex coal-like molecules, TMB, and then just metabolizing methanol over here. Okay, so very different in 13 CH3D and a little bit different in 12 CH2D2 at first as well. So sort of the takeaway here is that these methanogens were fed different substrates. and they produced different isotopolog distributions. But I kind of need to be careful here because chances are, isotopically, my TMB is probably very different from my methanol, which is probably very different from my MEOH. So how would I tell if this was, from the substrate or if it was from the pathway that is creating the methane from those substrates? And I don't have like a clear nail in the coffin answer for you, but I will tell you that because these are closed system incubations, in an individual bottle, the substrate is changing as the methanogen is processing that substrate. And you can see that in the methane product that it produces. So this is the delta C13, just the bulk isotopic composition of the methane from Shang-Lance is grown on MEOH. These are some of the replicates. And you can see, these ones over here where it's very light, maybe they didn't get through very much of that substrate, but then these over here, which had the same exact starting material, were probably able to get through more of it before I sacrificed that incubation, ended up with, something that was about 20 per mil different. And that's reflective of changes in the isotopic composition of the substrate. And what you can see, and similar story over here for the deuterium, what you can see is that the clumped isotope composition is not changing, even though the bulk composition is changing, pretty significantly during this time period. And so, a true nail in the coffin would be for me to run 2 parallel experiments where I have, 2 methanols with the starting composition is vastly different, and then check and make sure that they're still running the same clumped isotope composition. So that's not something that's been done yet, but this kind of stands in for them at this point. So I'm going to say, I think this is pathway dependent because the bulk isotopes do not seem to be affecting the clumped isotope composition. Okay, and so then what if I gave the same organisms, the same food, but I made them perform methanogenesis at different rates? And the way I did this is I had sort of my control experiments where, these were very happy methanogens. They were growing in the media that they liked at the concentration that they appreciated. They had, all the vitamins that they wanted and everything else. And then for sort of the experimental group, I diluted that media 5 times. And so they were basically living in an environment that was really uncomfortable to them. And as a result of that, if they made methane at all, and several of these cultures did not want to make methane for me, they made it at a slower rate. Okay, and so do these look different? And the answer is no. None of these are statistically distinguishable from one another. And each of these little dots here represents in between 3 and 5 of these cultures. So they are not different from one another. So the isotopolog distribution seems to be insensitive to the reaction rate. The starved methanogens produce the same isotopolog distribution as the methanogen millionaires, the ones that had everything that they wanted. But I will say with the caveat that the conditions that I was able to get them to still produce methane in the lab are probably still vastly different from what's happening in the subsurface. So they were producing methane at their most miserable, about 20 nanomoles of methane per minute, and at their happiest, about 55 nanomoles of methane per minute. So while these are very different from one another, they're also probably very, very different from what's happening, for instance, in the deep biosphere. And I could not get any of the hydrogenotrophs to grow at these really unhappy conditions. So food for thought. Okay, so to sort of sum up this part of the making molecules of methane, I'm really focusing on the metabolism as being the thing that's driving the clumped isotope composition. Potentially one could go to an environment, take a sample of methane we believe to be microbial, analyze it in this fashion, and be able to say, well, I don't know what this is, but I can tell you that it's performing, methyl atrophy, or I can tell you it's performing hydrogen atrophy, right? That was the idea, that's the plan, that's what I and many others set out to do. All right, however, when you do that, see we've made all of these really nice zones of methanogens in the lab, abiotic methane, low and high temperature, thermogenic methane, And then you take a sample of microbial methane, which cannot be any of these other types of methane from the Baltic Sea, and it ends up in a place on this diagram that I don't have a process to go to or to go with, right? It does not look like the microbial methanogenesis or any of these other things. So I'm going to spend the rest of my talk talking about how methane molecules are broken, because I think that is going to be a surprisingly important part of the methane clumped isotope story. All right, and so of course we know methane molecules are broken both aerobically, and this could be both a microbial oxidation, or it could be the type of oxidation that's happening in the atmosphere. Methane is interacting with chlorine or hydroxyl radicals. I'm not going to talk about that part of breaking methane molecules today. I'm going to focus on the anaerobic processes because I think those are the most important in the environment that I'll be looking in. All right, so I don't know if you guys can see this little Adobe Connect box, but it's in a bad part of my screen. It's driving me crazy. No, not going to let me move it. Okay, moving on, sorry. Okay, so we're here in the Bornholm Basin in the Baltic Sea. I'm going to show you now sort of the things that I've been talking about as pore water geochemical profiles. So we're out of the double isotopolog space. We're looking at everything as how it relates to sediment depth, essentially. So 0 is the sediment water interface going down below seafloor up to 30 meters in some cases. Okay, and so we have methane concentration. You can see it's sort of higher here near the sediment water interface, so we might infer that this is due to methanogenesis, and then as we move down core, there seems to be some sort of sink as methane is decreasing as we move down core. And then we have our two clumped isotope species, 12CH2D2 on the right, 13CH3D on the left. And this dark gray line here is similar to the curved black line from the earlier diagrams. This is thermodynamic equilibrium. So tied into this is the temperature of the subsurface, which is about 7.5 degrees Celsius constantly throughout the core. And so if methane was being formed in an equilibrium reaction in this material, it would all be on the line. And you can see that that is not the case. Up here near the sediment water interface, looks like we're pretty far away from thermodynamic equilibrium, but as we move down core, we seem to be approaching it. Okay, so I'm going to pull up here a few other properties. So this is the delta 13C and delta D of the methane. All right, and so something that is important to note about the Baltic Sea is that, I don't know if you guys can see, there's like these gray boxes on these plots. Those represent lacustrine sediments. So sort of during the last glacial maximum, as well as probably other glacial time periods, there's this huge Scandinavian ice sheet. And basically, the Baltic Sea was cut off from the Atlantic Ocean, and it was being fed by basically glacial meltwater. So we have these freshwater lacustrine deposits, and then sitting on top of them are brackish marine sediment deposits that look a lot more like the modern day conditions of the Baltic Sea. And so you can imagine that during the time you were depositing lacustrine sediments versus the times that these brackish marine sediments were being deposited, the organic matter and the water and its isotopes are going to be very, very different. All right, and so this kind of shows up in the bulk isotopes. So there's, for the carbon isotopes, for instance, where we're hypothesized based on the methane concentration profile that we are making methane. The methane isotopes are lighter, so that sort of goes in with this idea that biology will utilize the light isotopes first. And then we do have a sink, so as that methane is being consumed down core, we begin to leave behind an enriched residue, so that's good, that makes sense. But then suddenly, when we hit this boundary, things kind of turn around. And so, if we were just analyzing this in isolation, we would think, well, there must also be a methane source down here. There clearly isn't, or else there wouldn't be a methane sink. All right, so what's happening here? All right, and then the water isotopes are even messier, right? So there's, these very strange trends. It's very hard to tie that to what might be happening in a realistic situation. And this kind of gets back to that idea that with stable isotopes, the process matters, but also the substrate matters. And so this is sort of a substrate problem, right? How do we interpret this as people who just want to know, what the microbes are doing, what the microbes are eating? All right, so we're going to talk about a methane sink. Methane sinks. The anaerobic oxidation of methane needs a terminal electron acceptor. Sulfate is the one that we traditionally think of. So here's the sulfate profile. You'll notice there's no measurements from about the top 3 meters of this core. This part of the Baltic Sea was used as a munitions dump after World War I. So there is probably mustard gas canisters in this shallow part of the Bornholm Basin, so we were not allowed to sample that area, understandably. And so the upper sulfate profiles, we had to just assume that it was, close to seawater concentration at the sediment water interface, and then possibly quickly very decreased to get to these very low levels. And then there's sort of this little, bump in sulfate in the deep subsurface concurrent with this methane sink. So we are going to call this a sulfate methane transition zone. And then again, so because we have these lacustrine sediments, it's important to think about how freshwater deposits are going to be different from marine deposits. Freshwater deposits tend to be enriched in available iron, depleted in sulfur, as opposed to marine sediments or marine waters, which are going to be enriched in sulfate, but then not have a whole lot of available iron. So it's possible that iron is also acting as a terminal electron acceptor at depth here. And so my collaborators from Utrecht, Carolyn Slomp, and Matthias Egger did some pore water and sediment modeling, and were able to predict some rates of some of these processes that we're interested in. So using the sediment water interface concentration of sulfate to be like the seawater concentration of sulfate, we predict this big sulfate reduction peak here at the top of the core in this part that we're not able to measure, as well as methanogenesis concurrent with that just underneath it. And then finally in the deep subsurface here, perhaps a spike in iron AOM happening as well. And then because these sulfate concentrations are so low, they actually weren't able to produce with their model any sort of sulfate reduction occurring down here in the subsurface. So we have that sort of to work with. We think about this. So because we have these complicated, sediment issues, different, changing substrates, changing water, I want to focus in on the clumped isotopes to figure out what that can be telling us about what's happening here. And before I move on, I just want to say there were biomarkers present for this enzyme that we're interested in, MCR, and Me1, and then also relatives of the methanogen, methanohaladian investigatum. Okay, so all of that is present throughout here. Okay, so I did a very geochemist thing, and I made up a new parameter, which I call Psi12CH2D2. And it's very simple. Even I can do this math because I'm just taking the difference between the 12CH2D2 that I measure and what the 12CH2D2 should be if it's in equilibrium with the 13CH3D. Okay, and so in this very simple framework then, we can see if Psi12CH2D2 is 0, then that methane is in equilibrium. If it is at disequilibrium, then it is not equal to 0. It could be either less than or greater than 0. All right, and so here's sort of the same profile I showed you before, except we're looking at Psi12CH2D2 on the bottom. And at the top here, we are more than 6 sigma away from thermodynamic equilibrium. And at the bottom here, we're more or less overlapping with it through most of that area of the sulfate methane transition zone. Okay, and so one thing that we could think about is thermodynamic equilibrium sort of requires, when we're talking about something that's isotopically equilibrated, that means we have to be not just breaking molecules, but also making them. You cannot get to equilibrium just by consuming methane. There has to be actively making and breaking. There has to be this back and forth exchange to get us to this place, this happy place of thermodynamic equilibrium. So if you look at sort of the energy that's available to do either of these metabolisms, sulfate AOM or iron AOM in the subsurface, for the net reactions, you can see they are both negative, so they both should be going forward, but they are also pretty close to 0. And they're also pretty close to one another, so it's hard to say really here whether or not either of these would be more likely to move forward or backwards, but let's just say that they both have the possibility to have at least some reversibility associated with them, at least at some steps. Okay, so how could we equilibrate through enzymatic back reaction during AOM? And so here's a proposed pathway for sulfate AOM. It should look very similar to you because it is quite similar to methanogenesis. It's just running in reverse. And so we start up here with our methane, taking hydrogens off of it one step at a time, ending up with basically, CO2 at the end. But we also have right here, this first step, this first thing that methane sees is MCR, a homologue to the MCR that we talked about with methanogenesis. Okay, so what could be happening? Well, these experiments done by Yoshinaga et al. In 2014 showed that when concentrations of sulfate got low enough, below 0.5 millimolar, they could actually start to equilibrate the isotopes in methane with those in the DIC pool, okay? So you have to be able, if we're going to talk about reversibility, that means we need to be able to change the concentrations of either the products or the reactants and sort of drive this reaction in the other direction. And that is what an experiment like this is basically showing. And so this work was kind of summed up nicely by Timmers et al. In 2017 and some other work like it, and basically says that when we have concentrations of high sulfate, there is still some back reaction during AOM, but it's only about 3 to 7%. And during concentrations of low sulfate, particularly those that drop below sort of this magic number of 0.5 millimolar, up to 78% back reaction. So how I think about this is, in my silly, not an actual microbiologist way, is that to an anaerobic methanotroph, methane is food, yummy, get it in there, right? It's hanging out, it's with my enzyme MCR. This enzyme is a catalyst, right? And so there's an opportunity here for hydrogen isotope exchange. All right, but also as an anaerobic methanotroph, I'm living at the edge of thermodynamic life, right? That is really hard for me to do my job. I can't move all of this methane over that first sort of hump to go down further through this reaction, particularly when I don't have enough of my favorite terminal electron acceptor sulfate. So some of that methane essentially leaks back out of the cell after it's hung out on this enzyme, MCR. And in this way, you could actually drive a pool of methane in the subsurface towards this low temperature thermodynamic equilibrium. All right, it's a lovely story that I've told you. So how long might it actually take in this Baltic Sea environment? So I did some modeling using inputs like the rates that were predicted by Carolyn and Matthias, concentrations of various reactants and products, and I found that the time scale ranges from about 1,000 to tens of thousands of years in this environment, okay? And that's pretty appropriate for these sediments based on their age. The methane could have certainly been there that long or longer, so that's fine. And I did this model in 2 different ways. So the 2 lines you're seeing here, I just randomly selected this as an N member of being a representative of the methane made by methanogens in the subsurface. This is not necessarily, a realistic, but I had to start from somewhere, not knowing what that methane might look like. And the straight line you see assumes that it takes the same amount of time to equilibrate 13CH3D as 12CH2D2. But the curved line you see says, hey, wait a minute, we kind of have this problem where putting 2 deuteriums on a methane molecule can be hard, right? We saw that with the hydrogen tunneling in the abiotic methanogenesis. Maybe that's also happening here. And so that's the curved line that you see, which also tends to more closely follow the microbial array, which I will say is interesting, but it's just an observation. I don't have much more to say beyond that. Okay, so sort of to sum up the lines of evidence that AOM is driving this pool of methane to thermodynamic equilibrium. I have this carbon isotope reversal, which might be akin to what Yoshinaga et al. Observed in their 2014 paper. I have these really low levels of sulfate at this deep SMTZ, which are going to be much different than the marine levels of sulfate that are going to be up here. So this could potentially be spurring a big proportion of back reaction during AOM. The methanogenesis rates are decreasing with depth while the AOM rates are increasing. And then finally, I have this clumped isotope equilibration. All right. So what this means now is on a diagram like this, we would traditionally think of a process of AOM as that's just going to be something that's removing methane molecules from the system. In and of itself, it doesn't have a signature. It could move something along a pathway the way that I showed you would happen for microbial oxidation in bulk isotopes. But here, because we have this back reaction that's equilibrating, there's sort of actually like an end game for methane that's been interacting with anaerobic methane oxidizers here in this low temperature equilibrated space. So that's pretty exciting, and I would kind of hypothesize that we could take methane from any zone on the board, and if we were to feed it to anaerobic methanotrophs, it would end up here in this space. Okay, so I'm nearly done here. And so sort of the big pie in the sky take-home message is that, we've done this hard work of experiments and so on and so forth and figuring out where all these different end member, activities in terms of making methane and consuming methane will end up on this board. They're distinguishable from each other. We have maybe some beginnings of ideas as to why they are where they are. So that is sort of has some potential to be a part then of maybe the biosignature toolkit. And I could end the talk here on this really optimistic note in a Pollyanna fashion, which I definitely have been guilty of doing previously. But I'm going to end here instead, because I think it's important to acknowledge that natural samples rarely look like these end member scenarios, right? So this is all the natural samples that were measured at UCLA in the 2-year period from 2015 to 2017. So there's no experiments on here, no xenic cultures. These are just natural samples from the environment. And they're all over the map, and it's really hard to tie any of them to these specific end member scenarios. So I think, as we move forward, it's important not just to consider sort of this end member scenario thing, but all of these other processes, right? Natural environments are really complicated. Things are diffusing through Earth's crust. Things are moving through water. Reservoirs of methane are mixing with each other. And the clumped isotope composition of methane is going to reflect that environmental complexity. So it's important really to keep both of these in mind as we move forward, whether we're measuring methane here on Earth or hopefully eventually extraterrestrially looking at return sample methane from some of the other ocean worlds in our solar system, which is full of methane, so much methane. Excited about that. Okay, so stay tuned. This is still a relatively young field and an even younger proxy. I'm currently working on samples from the Ross Sea and the Corinth Sea, both of which are very different for different reasons from the Baltic Sea, for instance. And then in the fall, I'll be collecting some methane on IODP 385. And for the first time, I will have a companion in methane clumping with Didi Bojanova, who will also be sailing to do, in addition to all of the cool microbiology she does, also some methane clumping. So I want to say thank you, and thank you to C-DEBI for funding this work and making it possible. And I'm happy to take any of your questions.
Thank you.
Figure out how to get out of the screen.
You may want it up there in case someone asks a question. You could jump back to a figure or something.
Right. Okay. Oh, I'm stuck. I can't see the questions.
I don't see any popping up yet. There's a thank you for the great talk.
Okay.
Do you have the little chat box?
I don't. I'm like stuck on my desktop somehow. I don't know how that happened.
Okay. As they come up, I'll.
Yeah. there we are. Okay, now it's back. Okay, stop sharing.
Do you have any thoughts on the isotope space of methane produced via EET and or diet?
Yeah, that's a great question. I knew that. Hi, Una. I don't. I would love to measure it. Send me some. Okay.
Well, I guess that's it. Thank you very much for giving that presentation. It's an interesting field, and I look forward to seeing what you get with those samples, and more importantly, the ones from Mariana Forearch.
Okay, great. Thanks for having me, guys.
All right. Well, thank you very much.
Sure. Bye-bye.